Alright, I'd like to welcome you to this tutorial on Punnett squares. So to begin, we have to go over some basic vocabulary. So we have gene. A gene is just a character, and so like hair color, that is a type of gene. Eye color is a type of gene, and within that gene you have different forms of it. So like with hair color, you have such as in these pictures down below. So like you have different alleles for like a dirty blonde, okay, or copper red, or copper blonde. All these would have different alleles of that gene, which are just different variations. All right, and then the way you always write a gene, you have a dominant letter. So a dominant letter will always be capital T, or some type of capital letter. It doesn't necessarily have to be capital T, and then a recessive is always written in a lowercase. Now let's say in this case you go through Punnett squares and you have the letter S. Well, the letter S is kind of hard to distinguish between an uppercase and a lowercase. So in this case, what I always do, my lowercase, I always put a line above it. There's multiple different ways you can do it. In textbooks, you can see that lowercase is written in italics. You can put a line below it. You can put a line beside it. However, it helps you distinguish the difference between the upper and lowercase letter. Then you have phenotype and genotype. Phenotype is the physical characteristics, so you can physically see the color of my hair. That's the phenotype. But the genotype is the genetic makeup. What gives my hair color brown? Same thing in these cases. So what gives this person a dark brown? What gives this person auburn brown? We can see that the phenotype is that color, but what is the genotype? Right? And then when you want to write out the genotype, you write it again with these allele letters up here. So you have dominant T and recessive T. Right? And so there are two different types of homozygous. There are what I like to call homozygous dominant. Okay, and that's where you have two uppercase T's. Alleles always come in pairs. They never ever do not come in pairs. You can have homozygous recessive, so this would be two lowercase t's. And then finally, you would say what's called heterozygous. Heterozygous is where you have a dominant letter with a recessive allele. Okay, so again, homozygous dominant is where you have two dominant alleles. Homozygous recessive is where you have two recessive alleles, in this case lowercase, and then heterozygous. So these are just different types of Punnett squares. A Punnett squares just help predict a genotypic result of a fertilization between two different parents. Okay, so in this case we have a monohybrid here, which is just one gene. So we're just looking at seed shape here. Round seed versus wrinkled seed. Here, a dihybrid, so this is my dihybrid here, and this is where you're looking at two genes. So in this case, from the looks of it, we are looking at coat color of this mouse, and then maybe size, okay? Here we're just looking at two different genes, and you can tell the number of boxes increased greatly. Okay, over here we have 4. Over here it looks like we have 16. Okay, and then finally you have what's known as a test cross, still a monohybrid, but a test cross is when you're unable to determine. So again, we have those genotypes of dominant, and then we have homozygous dominant, homozygous recessive. Both of those would express the same phenotype, because dominant letter will always be overexpressed than what the recessive letter would be. So in this case, we would have to compare it with a known genotype, in this case, the homozygous recessive. And we'll do a couple of those problems. This is just the way that I do Punnett squares. This is the way I do it in my head. This is the way I would write them out. So this is just a little legend. I would first make a key. I would name the gene alleles. Then I go determine the parent cross. Then I perform the Punnett square, which is over here. Then I determine the genotype and phenotype offspring using a Punnett square. So I determine the ratios in that case, okay? Whether it's be like 1 to 1, 2 to 2, stuff like that. So let's start with monohybrid Punnett squares. We have different types of monohybrid Punnett squares. So in this case, we have a couple human type Punnett squares. So we have tongue rolling, which is dominant. You have free earlobes or attached earlobes. Free earlobes is considered dominant. Hitchhiker's thumb, all right, whether you can bend your thumb back, which is known as hitchhiker's thumb, that's dominant. You have facial dimples, and then you have widow's peak. So here on the pretty much the hairline, whether your hair dips down, in this case, the lady to the left here would have widow's peak. All right, so let's start with a problem. This is a simple monohybrid problem. You just simply start by going through and reading it. So we have in tomatoes, red fruit is dominant over yellow fruit. So remember my key, the first thing I like to do is make a little legend. So I know red fruit is dominant and yellow fruit is the recessive. So I'm going to use capital R for red fruit and recessive R for yellow fruit. And that's just making a key. So I name the gene, which is fruit color, and then the alleles, red, yellow. Okay. Now we do the parent cross. So design a cross, okay? Design a cross between a homozygous dominant. So it says homozygous dominant. So because it says homozygous dominant, I'm going to do 2 capital R's. And then with a heterozygous plant, all right? So that's going to be a capital R with a lowercase r, okay? And then I just put in the corresponding information. The way I always do it is that I put the first parent up at the top and then the second parent down the side. In most cases in genetics problems, usually the male would go up top and then the female down the side. But as long as you remain consistent, you're all right. So here we have a homozygous dominant genotype. Here we have another homozygous dominant genotype. Here we have a heterozygous genotype and another heterozygous genotype. So right now we need to determine the ratio. So the phenotype, remember, is what you can physically see. We can physically see red and yellow fruit. So we have red versus yellow fruit. Okay? So if we count here, this is going to be red, this will be red, this will be red, because remember the dominant allele is always expressed over the recessive, and this will be red. So we have a 4 red to 0 yellow, and if we just reduce that, it'll just be 4 red. Okay? Then the genotype can be, remember in a monohybrid there can be 3 different types of genotypes, okay, if it's possible. So we can have homozygous dominant, we can have a heterozygous, and then we can have homozygous recessive. So if we look here and count how many homozygous dominants we have 2. All right, so we have 2. And then if we count how many heterozygous, we have 2. And then homozygous recessive is 0. And then if we just reduce this, it'll be 2 to 2. Homozygous dominant versus heterozygous, and then you just make it 1 to 1. Okay? All right, so let's move on to another problem. This is a test cross problem. So again, a test cross is where we have an unknown genotype. So in this case, in cattle, black coat color is dominant to white coat color. So we know black, so we name the gene is coat color, and we know that black is dominant, so we're going to put a capital B there, and then a recessive B there, okay? And a farmer has a black male of undetermined genotype, so he does not know if it's a homozygous dominant or heterozygous. He doesn't know. Okay, so how can a farmer determine the genotype of the male? Well, that's very simple. A homozygous dominant, all right, and then a homozygous, or heterozygous, I'm sorry, heterozygous, okay? And then we just cross it with a known genotype. So we know the known genotype is going to be white coat color, and that will always be a homozygous recessive in this case. Okay? Because the only way you can ever see a recessive trait is if it's homozygous, if it's paired up with another recessive trait. So in this case, let's go ahead and form the Punnett square. So all of these are going to be like this. Going to be all heterozygous, and then here we'll come out with 50% black and 50% white. So if we just do the ratio, so the phenotype for this one So we have black versus white. Okay. In this case, are we going to have any white? So we're going to have all 4 are going to be black. Okay. And then if we look over here, we're going to have black versus white. All right. So we have 2 black, as we see here. These are 2 black, and then we have 2 white. So it'll be 2 to 2, and then we just reduce that to 1 to 1. Okay. And then if we look at the genotype ratio, so this is the allele letters here in the middle. So if we look at that, we have this. So this is going to be 4. And then if we look at the other one, we have a heterozygous and then a homozygous recessive, which in the end, if we count up the number, it's going to be 2 to 2, which in turn you can reduce to 1 to 1. Okay. So this is a test cross, and you only do this if you have an undetermined genotype between a homozygous dominant and a heterozygous. Okay. These are a couple practice problems. So you have problems 1 through 4. 4 is a test cross. So this is where you are given the information, but you do not know if it's a homozygous dominant or heterozygous. Okay, and I just want you to show your work. Make sure you show your work. Use the steps that I showed you, so that we can go over these in class. Hope this helps. Have a great day.